Randomization. It can sound technical, but in the world of clinical trials, it's absolutely vital. When allocation is left to personal judgment, subtle preferences and clinical impressions can influence who receives which treatment, introducing selection bias. Randomization replaces judgment with a pre-specified chance mechanism, making the comparison as fair as possible and supporting blinding. It helps ensure that any differences seen at the end of the study are more likely due to the treatment rather than who was enrolled into each group. So, what is randomization in clinical trials? Simply put, it is the process by which the study team assigns participants to different treatment groups, using a method based purely on chance. The key features are allocation concealment, so upcoming assignments cannot be predicted, and adherence to a written randomization specification. This might sound simple, but it's the backbone that keeps clinical research objective, trustworthy, and robust. It also aligns with ethical equipoise, when there is genuine uncertainty about which option is better, and it underpins common statistical methods that assume random allocation. Let's break down the core approaches. First up, simple randomization. In practice, systems use a random number generator to give each participant a predefined probability, for example, 1-to-1, of entering each group. This works well with large trials, since numbers tend to balance out over time. In smaller trials, however, pure chance can lead to temporary imbalances in group sizes or key characteristics. But what if your trial is smaller, or you want to make sure groups stay balanced as you go along? That's where block randomization comes in. The design uses blocks, prearranged sequences compatible with the target ratio, for example, blocks of 4 or 6 for a 1-to-1 trial, so that after each block, group counts remain close. Varying, undisclosed block sizes reduces predictability at sites and protects blinding. Now, stratified randomization. When baseline factors such as site, age group, or disease severity are important, the plan randomizes within strata defined by those factors. This balances them across arms, improving precision and interpretability. Care is needed to avoid too many small strata, which can hamper enrollment or create operational complexity. Occasionally, design teams use unequal allocation like 2-to-1 or response-adaptive methods. Reasons include gathering more safety or exposure data on the experimental arm, addressing cost or supply constraints, or ethical considerations. The trade-off is statistical power. Moving away from 1-to-1 often requires a larger total sample to maintain power. Adaptive methods adjust assignment probabilities based on accumulating data, but they must be pre-specified, independently monitored, and carefully analyzed to avoid bias. Here's the key. Randomization isn't just an administrative step. Planning is everything. A robust plan specifies allocation ratios, allowed block sizes or ranges, and stratification factors, with a short rationale for each. Multi-center trials benefit from centralized systems such as interactive response technology to apply rules consistently, maintain audit trails, and prevent prediction of upcoming assignments. Roles are separated so that schedule generation and access are independent from data analysis and clinical teams. Before go live, teams review dummy schedules to confirm formats, kit numbering, and data flows. Confidentiality is protected through restricted access and encrypted transfers, and an emergency unblinding procedure is documented for participant safety events. Ongoing quality control checks allocation counts overall and within strata, with thresholds that trigger investigation, and all specifications, code, outputs, and QC logs are archived for audit readiness. But there are pitfalls to watch out for. Common issues include treating randomization as a formality, choosing blocks or strata without considering enrollment patterns, allowing predictability in block sizes, skipping routine monitoring of group counts by site and stratum, and weak documentation or archiving that makes audits difficult. Establishing clear thresholds for review, for example, predefined tolerances for imbalance and acting early, prevents larger problems later. In summary, well-planned and well-executed randomization improves fairness at allocation, supports blinding, and strengthens the credibility of trial conclusions. Careful design, secure operations, and routine monitoring are the essentials that keep it working as intended. If you'd like to learn more about how Quanticut can support your trial, our teams have extensive experience in designing and implementing randomization strategies, including centralized systems, unequal allocation, and multi-center setups. To explore how we can tailor our approach to your study needs, email us at inquiries@quanticate.com or submit an RFI through our website.